viewers. I know it's been a while since my last video, but I've been busy preparing for my qualifying exam. In doing so, I realized, hey, can't I just read some of the stuff that I'm reading for my qualifying exam as part of my channel? So, here goes. Today I'm reading an article entitled Normal Stem Cells and Cancer Stem Cells, Similar and Different. I've been reading it for a while, so I'm just going to scroll down to the bottom and continue with my reading. So basically, just to summarize, this article is about, well, as you can see, normal stem cells and cancer stem cells. The prevailing theory right now, one of three actually, is that cancer is a function of stem cells, or cells that function kind of like stem cells, but themselves are not exactly stem cells. But you can kill the downstream cells that are generated from these cancer stem cells. But if the cancer stem cells don't die, then you can still get cancer redeveloping within an organism. So this article is just a comparison of those kinds. As you can see here, there are three different theories. There's the cancer stem cell model, there's the clonal evolution model, and then there's the interconversion model. Let me know if you'd like to hear more about these kinds of things. Sorry about that. So anyway, here's where I've been reading. Interconversion in normal stem cells and tumorigenic cells. The ability of some normal tissue stem cells to interconvert between different functional states is important for their roles in adaptive homeostasis. Cell labeling studies have suggested that highly purified mouse hematopoietic stem cells, HSCs, may interconvert between dormant and activated states in conditions of hematopoietic stress and recovery therefrom. Notably, CD34 expression increases in murine HSCs during conversion to an activated state and reduces after resumption of steady-state maintenance, indicating that functional interconversion of stem cells may be associated with phenotypic interconversion. In the skin, different populations of epidermal stem cells display different degrees of multilineage differentiation depending on the requirements of the organ. For example, in steady-state maintenance of skin integrity, the interfollicular epidermis is maintained predominantly by interfollicular epidermal stem cells, and the hair follicle, HF, is maintained predominantly by HF stem cell populations. However, during healing of epidermal wounds, HF stem cells temporarily contribute to repair of the interfollicular epidermis, and hair follicle neogenesis is newly repaired skin can derive from interfollicular epidermal stem cells. These studies indicate that normal stem cells may change their proliferative and differentiation fates in response to epidermal cues. The ability of at least some normal stem cells to interconvert between different fates depending on the requirements of the organism provides great adaptability and homeostatic maintenance. It is likely that tumorigenic cells in at least some cancers can interconvert between different malignant fates and even different states of sensitivity to therapy. For example, cell cycle studies of leukemogenic human CML cells showed heterogeneously quiescent cell populations. Yeah, sometimes even scientists struggle with scientific jargon. Anyway, for example, cell studies of leukemogenic